Good. So I thought Jen from the Friedreich Ataxia Research Association, they've been very successful over more than a decade of really establishing here a platform bringing all stakeholders together. In very many ways, I think a role model for us on how to incentivize different stakeholders to take an interest in a rare disease and move things forward towards trials, move things forward in terms of understanding of the preclinical space of basic science. So, Jen, please.
Florian, thank you for having me here, and Amber, thank you for also bringing me here. This has been great for me. I've left with a page of notes of ways in which we can collaborate together. I didn't realize ahead of time how much we had in common. And yesterday, talking about outcome measures and biomarkers, there are more of these research studies that we're doing in common than not. And so for me, one of the takeaways is that, you know, for a lot of these studies, the more we can share information about protocols. I think yesterday one of the examples was the MRS protocol, coming up with a way to make that multi-site, to make it something that can be implementable in clinical trials. This is the same issue that we have. And so that is a perfect example of how our researchers and our organizations can work together to solve a common problem, because it's not unique to each individual disease. And as advocacy groups, those biomarkers, we need to get qualified with the FDA. One of the comments from industry yesterday was one of the things they're looking to us to do is help figure out the regulatory path. And one of the ways that we do that is by working with the FDA to educate them about our disease and to work with them to qualify endpoints and biomarkers. And if we're going in as 3 or 4 diseases and saying this technology or this outcome measure, or this biomarker works in 5 different diseases and could be a qualifiable endpoint, we're going to be so much more powerful in doing that and having our voice heard and having those meetings and getting those endpoints approved. And so I think this is a real place where we can make a lot of progress together. I did prepare a little bit of background about FA and about FARA, just so that you have some kind of idea of how we came to be and what kind of an organization we are. So this is a picture of 3 young boys, Byron, Keith, and Stuart. And FARA's founder, Ron and Rachel Bartek, their middle son, Keith, was diagnosed with Friedreich's ataxia at age 11. And it was right around the time when the gene was identified. So the gene was identified in 96, and Keith was diagnosed in 97. And when Keith was diagnosed, Ron, when he tells the story, says, you know, Rachel and I huddled around the computer, started to look for information about Friedreich's ataxia, and there was precious little, and what we did find was absolutely horrifying. They live in Washington. They're lobbyists and state people. Rachel worked for Billy Tozan at the time, and so they started marching into the NIH, like, can you help us, can you help us? And they were told, no, like, you can help us because there is no research organization devoted to this disease. There's nobody promoting research activity, and that's what you can do. And so Ron and Rachel founded FARA in 1998, along with several other patient families, and importantly, also with the scientific community. And that's what I hear here with ALD Connect, is that this is advocacy and research coming together. And that's what Ron and Rachel envisioned from the very beginning. They wanted an organization that was solely devoted to advancing the research and building a research community. And they did that with 3 scientists at that time who they found who were working on FA, and there really weren't many. And, you know, it was the identification of the gene, I think, that really brought those people together at that time, that at least gave a door to open. And it sounds like that's what you have at this time as well. There's a lot that's been learned about the disease, the underlying causes, and there's more opportunities now to think about how to advance therapy. And that's really what brought the FARA group initially together. Anybody who's met Ron Bartek knows that you cannot get within 2 inches of him or 200 yards without getting hugged. Okay, Ron's a hugger, but collaboration and partnership are the cornerstone for our organization. Ron says, we partner with everybody, and he absolutely means it, and he means it physically as well. He will hold you there until you find a way to work together. It is true that we do have to focus on Friedreich's ataxia. Not everything we do is about all diseases. And so to Florian's comment earlier about, we also have to focus on our disease, yes, absolutely. But there are a lot of common issues that we can solve together. And if we do that, we'll be much more effective. And Ron saw this early on, and it's definitely a part of our culture that we cannot afford confrontation. We cannot afford isolation. We won't make progress in that way. The other core principle of our organization is similar to what I've heard here, which is urgency. Time is not on our side. Individuals with FA have an average life expectancy of 30 to 5 years of age. In addition to it being a neurodegenerative disease, it's a metabolic disease, and it affects the heart, and it causes life-shortening cardiomyopathy. So our strategy early on was to really grow the scientific community. Ron will tell you, he would recruit anybody who was even willing to try and say Friedreich's ataxia. You didn't even have to know how to spell it. We'll talk to you. But we also know that, when you think about treatments, you're not going to be just one and done. You're going to need a diverse, deep treatment pipeline. And that's something over the last 10 years we've worked really hard on, thinking about how to have a diverse and deep treatment pipeline. We knew we'd need to organize the patient community. Research is only going to go so far in the lab, and our community needs to be mobilized for research as well. We knew that public-private partnership was also essential. FARA was probably one of the first organizations to actually give funding and an investment to small biotech startup. We made our first grant to a company back in 2004. And we continue to look at, funding isn't the only way we partner, but there's lots of different ways we partner, but we knew that this was a key part of the equation. And then obviously advocacy is a big part of what we need to do. So we have a board of directors that is made up of patient families, academic researchers, industry researchers, business leadership, people with expertise in communications, legal. Some of the folks on our board have absolutely no personal connection to FA or any professional interest in FA, but they've got an expertise, let's say, in communications that we need. And so our board is really dynamic, and I think it's one of the real strengths of the organization. It's a big board. We have 18 people on our board of directors, which as the executive director is interesting to manage. But I think it's important because as a nonprofit, and we rely a lot on volunteer support, and we have a very active board. This is not a rubber stamp board. This is a working board, and it's always been that way. Same thing with our scientific advisory board. It's both academic and industry based, and we ask them to do a lot. It's not a named board. These folks are on conference calls once a month, sometimes twice a month, reviewing grants, reviewing proposals, helping us design new programs, reviewing our pipeline, all different kinds of activities. And I was the first staff member hired to FARA. I was hired in 2006. And we now have 7 full-time employees. Most of them are doing research most of them are doing fundraising and communications. So in my office of 6, I'm the only one focused on research. Everyone else is focused on fundraising and communications. A lot of our support for our research program is we have 2 part-time people, PhD folks who help me run our research programs, and they're remote. And then Ron is the heart and soul of our advocacy effort in Washington. So the gene was identified in 1998, and in 2004, this is what the research pipeline, the treatment pipeline looked like. And over here are different mechanisms of action. So here would be compounds that try to reduce oxidative stress and increase mitochondrial function. In FA, the gene with the mutation and the protein that it codes for, while it's a nuclear gene, the protein functions in the mitochondria, and it's a protein deficiency. And so when you don't have enough frataxin, you have oxidative stress and mitochondrial dysfunction, and I'll spare you all the details why. But this was one of the things that we learned shortly after discovering the gene. And so trying to improve mitochondrial function or reduce some of the consequences of frataxin deficiency has always been a therapeutic approach that we've looked at. Again, part of the function of the specific gene and what happens in the mitochondria is that there's an increase in iron in the mitochondria, and so we've looked at ways to try and get rid of that iron. And obviously we've tried to increase frataxin expression as another therapeutic approach. And these were kind of all of the ideas that were going in 2004. At that time, FARA's research program was really starting to take off. One of the first things FARA put in place was a grant program, and so we fund traditional research grants, basic translational clinical research. We accept grants 24/7, 365. We have moved to deadlines because it's a little crazy to manage, over 50 proposals coming in a year. But the board has maintained that if you have a really great idea, we don't want you to wait 6 months for a deadline to submit it to us, so send it to us anytime. I need to update this. We're now over about $35 million in research funding since we were founded. The past few years, our research funding, because of our fundraising efforts, is now up over $4 to $5 million a year. And when I started in 2006, we were at about 500,000. So in 10 years, we've gone from 500,000 to close to 5 million in research funding each year. When I was brought in 2006, one of the first things Ron said to me was, Jen, I need you to start a patient registry. We have an Excel spreadsheet with people's names on it, but from what I'm hearing, that's not a patient registry. And so can you start a patient registry? And so that was the first thing I did. It's an online registry. Patients enter their own information. So it's international. And since 2007, when we launched the registry, we've had over 2,600 people register. We currently have about 2,400 active registrants, and just over 1,400 of them are based in the US. We use our patient registry to recruit for clinical trials and clinical research studies. It's not so much a research tool, the registry. It's more of a recruitment tool for us, and it's been really effective. I announced a study in June for a trial enrolling 90 subjects, ages 10 to 25. And so I could go into the registry, pull out people living in the US, ages 10 to 25, who have a certain ambulatory status, and email them all the information about the study. Within 4 hours of sending that out, one of our sites had a list of 50 interested participants, and they had 30 slots that they were supposed to fill. And so, they had good qualified candidates already on their list. It's been really effective. We have not had a clinical trial fail due to lack of enrollment. And this is our primary tool for enrolling patients. It's also been really helpful to figure out where people are. You know, this was another comment we heard yesterday from the industry panel. We need to know that you know where the patients are. Well, it's now like on our website. You can see, you can go, it tells you what states, what countries people are in. We've learned since launching this registry that, you know, we have over 200 patients in Brazil. We have hundreds of patients in India. These are places for us to, you know, grow our global community. But it also helps us get the interest and the support from our industry partners, because we can show them evidence of where the patient population is. Prior to joining FARA, I worked at Children's Hospital of Philadelphia with Dave Lynch. And I was clinical research coordinator and genetic counselor working with him. And in 2004, we started a clinical outcome measure and natural history study. And it was joint funded by FARA and the Muscular Dystrophy Association. And that was for the first 3 years of this study. And then FARA took over funding the study, and I'm still the overall coordinator of this study, but it's grown into a much larger clinical research network. But the backbone of it is a natural history study and clinical outcome measures that we started collecting 11 years ago. We now have over 820 patients. They're all seen every 12 months with the same battery of tests that we started doing 11 years ago. We've added new tests. We've taken only one or two tests have come out, but we've added some new tests over time. But we now have that natural history data. You know, the original cohort of 70 people who were enrolled, we now have 11 years of data on, and we can show you on certain measures how much they change every single year. And we're using that data now to design our clinical trials. How many patients per arm for this particular measure? We have the data we can pull. The frustrating thing about this is it's never, ever good enough. My advice to you is just start, and you've done that, right? But you're always going to want a better outcome measure. You're always going to want a better biomarker. You know, some of the outcome measures we don't like because you still have to do a 2-year trial. But at least we know and can walk in confidence with, if we use these measures, this is what we have to do. In the meantime, we've been looking for new and better measures. And so you have to be flexible. You have to constantly run some side pilot projects of new measures, and and let some of them fail, and then the ones that look good, then bring them into the whole network and move them forward. We have a scientific conference program. About every 2 to 3 years, we bring the whole research community together for 2 or 3 days and cover everything in FA from soup to nuts. This year, we did this in collaboration with our European Friedreich's ataxia and all ataxia groups. And so we had an international ataxia research conference. It wasn't just FA, it also included other hereditary ataxias. And Ataxia UK, a group in Italy, a group in Ireland, and FARA all sort of collaborated in organizing and hosting the meeting. We had the largest research conference ever for hereditary ataxia. We had over 350 scientists. We had more than a dozen drug companies participating and presenting data at the meeting. It was really a big accomplishment for our global community. And when we talk about advocacy, most people think of government-based advocacy, and this is certainly a part of what we do. But I think advocacy cuts across different things, and a big part of our advocacy effort is that we have to partner with other organizations. We work with MDA, we work with Cystic Fibrosis Foundation, we work with NORD. All of our advocacy efforts are joint partnerships with other groups. And so I don't know if you've seen this slide before, but there's this clinical trials transformation initiative you'll probably be hearing more about, especially in sort of the rare disease circle in Washington. And the CTTI is coming out with a whole series of papers on patient engagement in clinical research. And so this is borrowed from them, these chevrons, and kind of highlights different ways in which we can be engaged in research through the whole process. And I won't go through this in detail, but what FARA's tried to do is pick some of these in each of the boxes and say, okay, what are the things that we can do that are going to help advance and push us through this pipeline faster. And a lot of the things we've chosen have been the things that fall into that pre-competitive space. If we can fund a better animal model, that helps all projects. Our clinical network, our natural history, our patient registry, all of those things we consider pre-competitive. And if we can share those resources across whatever the program is that's coming through the pipeline, that's gonna be the best place for us to put our effort. And so this is what our treatment pipeline looks like now. We've had 3 drug trials launch since the beginning of this year. And so we're currently enrolling about 200 patients into clinical trials this year. Some of these drugs that are in later phases, some of them are finishing up trials, but 4 of them are active, open enrolling studies right now. And while this is really great, we know that the odds are not in our favor and that many of these will not work. And so we still maintain a really active drug discovery program. A lot of our basic funding, a lot of our basic grant funding goes to early stage drug discovery research. We need to continue to fill this pipeline with new compounds. And so this is just a little picture that I put together last year because we're working with different stakeholders, right? We have our researchers, our physicians, our patient families, our donors, industry, government, other advocacy organizations. And what FARA is doing, what ALD Connect, what your individual organizations are trying to do is pull all of these together. And for me, success for FARA as an organization and for FA as a research community, when we're effective in doing this and everybody's right here in the middle working together, we're making the most progress. That's when we're able to do our best work and be effective. Anytime we're out on the edges, we're probably not being efficient and effective. And so I just continually view it as our role to bring all these stakeholders together as often as possible and promote that collaboration. So I'll stop there.
So it sounds like almost you have to now put a larger ring around that ring to include interaction with other diseases, right? But that was fantastic. I guess 2 quick questions as we're sort of looking at our pipeline and how we want to advance. And we spoke yesterday briefly about how much information do you need, how much data do you need to proceed, right? And do things go sequentially or do they go in parallel, right? And do you need the perfect biomarker before you can launch the trial? And I guess my question here, as you were gathering natural history data, how much of that was informed by pure natural history studies versus first trials that also were, adding placebo or adding additional information, or failed trials that you might think are also data that you might want to incorporate? Maybe you can speak to that briefly.
Yeah. So actually, the one thing I will tell you is the companies coming into the space now to work in Friedreich's ataxia. When we sit down and say to them, "Hey, why are you interested in this disease?" It's because of the patient registry and the natural history study. That's the answer I get from them. because of that, they can put together their story about what the regulatory path might look like. It might not be the best measures that they want to use, but at least it's something to go on for them to start. One of the interesting things I've learned is when working with a lot of companies, they want to be able to, on paper, map out the plan to approval and know exactly what their phase 3 study is going to look like before they even do a tox study in animals. That was a huge learning experience for me. And so that natural history study, we started that in 2004, and it was actually because one of our investigators stood up at a 2003 conference and said, hey, you know, this vitamin idebenone people are looking at, but we have no way to measure what's going on in patients. Like, we need to have outcome measures. And so he developed a rating scale, and there were a few other rating scales that had been developed in Europe at the time. And so the clinical investigators came together and selected a clinical rating scale. But then they also looked to other diseases. And in this case, we looked at multiple sclerosis because at the time they had made a lot of progress in performance measures. And there was a lot of overlap in some of the things we'd want to measure, like gait and hand performance, speech. And so we actually borrowed from MS all of their functional composite measures, and we used their quality of life. I noticed you were using that as well, the MSQLI. That's the quality of life measure in our natural history study. It was validated in another disease. There was a lot of data from general population, from controls, and so that was where we started. And unfortunately, over time, we've added things as trials have evolved. So we've stuck with that basic core for a lot of our clinical trials as outcome measures. And then as a new trial has come up and a company has an idea of, hey, we should look at glucose tolerance and insulin resistance as a biomarker. You know, we went flying into that study with no preliminary data. So we've done some of that, but that has changed what's been added to the outcome measure in natural history study over time. So I think, does that answer your question?
But it's also worth mentioning you have all that serum stored. No, it's worth mentioning that you have all that serum stored, so if later we want to look back, you can like pull those samples.
Yeah.
That's huge.
So in the natural history study, those 820 people who are enrolled, every time they come in for a visit, we draw about 8 tubes of blood. We've got DNA on every person. We have RNA on every person. We have RNA on some people up to 5 years now. We have serum, plasma. We've stored all of it. And some of it we've used for studies over time. But to Amber's point, yeah, Amber came in and said, hey, you know, has anybody looked at troponin? And so, you know, we can pull 50 samples out of the freezer, look at troponin, come back and say, okay, do we have something here? Do we not? And that has been a huge resource. We've also put a fibroblast repository into place now as well. We have over 50 fibroblast lines growing and available for the research community.
So in these clinical trials, how do you define what's a meaningful change?
So Kihan asked a good question. What's a meaningful change? So through lots of discussions with the FDA for some of the trials that we've done, there are things in our clinical rating scale that they will agree are clinically meaningful, like how long you can stand. And so the FDA has helped us go through our clinical rating scale and select for all of the subgroups that are clearly, directly, obviously clinically meaningful. And then the ones that are not, like whether or not you have reflexes, the FDA doesn't think that's meaningful, whether or not you have reflexes. Take that out. And so we've actually modified that rating scale over time. We've also correlated a lot of this with ADLs and quality of life measures. And so we do have some of that data as well. The FDA has told us that, that quantified neurologic exam, as long as it's subgrouped out into the things that are directly functional, that's good enough evidence for them for a phase 3 endpoint.
So this is a discussion with the FDA you should have prior to...
Yes, these are discussions that you can have with the FDA prior to starting clinical trials. And I can't stress that enough. Make friends with the FDA. Invite them to these meetings. They will come. We had a clinical meeting in June of 2003, and it was just to review all of our natural history data, all of our clinical outcome measures. It was 2 days of just all the clinical data we have from the US and Europe. And we held the meeting in Silver Spring, Maryland. Key, you have to have the meeting in Silver Spring. But we had 11 people from the FDA come. So if you invite them, they will come, but you do have to make it in their backyard because they can't travel.
I wanted to ask just a question about samples. I'm here.
Oh, sorry.
How, where are your samples actually housed, and how do you sort of deal with the logistics of different investigators with their own data banks and things like that?
Yeah, great question. I was a little jealous yesterday. One of the things about starting 11 years ago was that some of the nice technology that's available now for sample tracking, and we don't have any of that. And so I sent my PI an email yesterday, we really need to up our game with sample tracking. So they're in 3 different academic labs. And this is kind of the collaboration thing, right? We have really good relationships with those academic collaborators. And so we can go to them and say, hey, Pfizer needs 50 samples because they want to do X, Y, and Z. And you do need to put a material transfer agreement in place between the company and the academic institution. Once that material transfer agreement's in place, the samples go out. The study has to be scientifically valid, but the investigators keep a pretty low bar in terms of gatekeeping over the samples. We can do that, though, because every year we're replenishing the stock. That was something that was discussed yesterday. When you have samples where you're not replenishing the stock, that's a harder issue. But for samples where, like serum and plasma and DNA, where we can constantly be replenishing the stock, we don't have to set too high a bar on sharing. So
in that regard, I would say that the example we should follow is the International Brain Tumor Consortium that has a biorepository that's worldwide. And I don't know how it's handled, but I know people in Rome can access it. They have the clinical histories, they have the MRIs, and I think that's a good model to actually look at.
Yeah, we have a researcher in Albany, a neuropathologist who has run a tissue bank for us. He runs an autopsy program and tissue bank, and he has samples from over 40 patients. And so his bank is large enough to meet the needs of the research community, and so he runs it the same way. I mean, he shares samples with anybody at this point who needs them.
How much is of your outcomes that you're reporting are patient-reported outcomes, and how important was it for you to have patient portals or other social media in terms of establishing engagement, or is it all about just trials and announcing them? And maybe you can shed some light on that.
So I was having a flashback yesterday when you said 500 or 700 people on Facebook page. I remember when we hit our 500 mark, and we were really excited. We couldn't believe that there were that many people out there. We're now at 7,500, and our average post gets shared to and viewed by over 25,000 people. So stick with it. It's critically important. I actually think that people get my trial information faster on Facebook than they do through my patient registry. Facebook's been, and social media are absolutely necessary to engaging the patient population. And it's been a great way for us to explore some new issues with the community as well. We are now, in terms of patient-reported outcomes, we've always had a standard set of quality of life, activities of daily living measures, but we're now looking at some of the other things that you're looking at in terms of wearable devices, cell phone apps. We have a cell phone app study right now that we're doing as a pilot to measure the amount of It's an app where you can go through a series of 5 tests. One's a walking test, one's a balance test, one's a speech test, and then 2 hand coordination tests. And you do that 3 times a day. But then when you're not doing those 3 tests during the day, the app's collecting information about how much you're walking, just in general distance, how many times you're going in and out of your house in a given day, how often you're just interacting with your phone, how much activity you're doing, and hoping that these will be other ways for us to collect patient-reported outcomes.
Is telemedicine of any value?
Telemedicine, I think it has to be. So we have 8 centers in the US. One of them's on the West Coast. None of them are in the middle of the country. And our average patient travels over 650 kilometers to one of our clinical research network sites. And one of the studies we did over the summer was in the patient registry. We queried everyone who we've sent clinical trial announcements to and asked them questions about why they choose to participate and why they don't choose to participate. And after not being eligible for the study, the second reason why people do not participate is because of travel. And so we have to be able to conduct some of these measures in the home. We have to reduce the travel burden. It's not just about paying for travel for people to get places. It's about reducing the burden of travel. Because we're not going to be able to have centers in every state and every city for rare disease. That's just not going to happen. One of the home-based measures I just looked at that was really neat was using Microsoft Connect. So they've done this in ALS and in Duchenne, where you sit in front of the computer screen. The computer's on the desk, and on top of the computer is the Microsoft Connect device. Anybody with kids knows that that's like the Xbox gaming thing. There's one in my basement. I didn't even know it. It knows my son really, really well. It doesn't recognize me or my voice. But when you sit in front of the Connect device, you load up this computer program, and it tells you, put your hand at your side, move your arm straight out in front of you, return your arm down, then go to the side. And they're looking at how much you move in a workspace and how effectively you can move in that workspace. And so in muscle diseases, you can imagine you're not able to complete the range of motion, and so you're not using the total workspace, and they can quantify that. In ataxia, you might be able to get your arm out, but how you get there is not efficient. And so these are all measures that you can do in your home. So those are the kinds of things also with, I think, telemedicine that we can be looking at together. Yep. Oh.
Thank you. I have a practical question regarding self-enrollment in your registry.
Mm-hmm.
How do you make sure that people who self-enrolled really have your disease?
So we ask a series of questions, certainly around for symptom and around genetic confirmation. So genetic confirmation's been available since 1997. We also ask about other family members. So we ask several questions, and then when the data comes when a new patient registers, it actually comes in, and the person's not into the registry till I qualify them. And I go through the responses and make sure that they make sense. Occasionally, I will get somebody who tells me that their mom or dad had Friedreich's ataxia, and then that elicits me to go back to them and say, mom or dad had ataxia. Are you sure you have the Friedreich's ataxia? Do you have spinocerebellar ataxia? Oh, yeah. You know, and so we try and curate the data as much as we can, but it's not perfect. And for our patient registry, that's okay. For our natural history study, you have to have genetic confirmation to be enrolled in that, and that's physician-entered data. And so having the two, we can be a little bit more lenient on the patient side.
Quick question about consent. So for the patient registry, do you consent them? I understand you have to consent for the natural histories, but what about the registry? How can you use the data from that?
We do have an online consent that you have to go through before you enter your information. FARA has held that, so the data in the patient registry is FARA's data. We don't share identifiable data outside of that, and that's what we've said in the consent form, and that's what we've done. But what we'll do is if there are requests for analysis of the data or for us to push something out to the registry, we've been able to do that.
Do you use any commercial IRB, or how do you? Who approved your consent?
So we have not. So we're actually, because we're not using this primarily as a research tool or a medical tool in any way, where we just get assent.